Making a single, precise cut requires the brain to activate the specific motor plan for the scalpel stroke, while simultaneously suppressing thousands of competing, unwanted impulses that would otherwise cause a tremor. This filtering process is handled by the basal ganglia. Unlike the primary motor system, these structures have no descending tracks to the spinal cord. They function as an accessory, parallel processing loop that modulates motor signals within the brain itself. In its baseline state, the thalamus is tonically closed. It constantly attempts to excite the motor cortex to initiate movement, but it is held back by continuous inhibitory signals originating from the basal ganglia. This means initiating movement is not a matter of simply applying power. Instead, the brain must selectively release the physiological brake on one specific plan while keeping it clamped shut on all others. Mapping this control system requires a look beneath the cerebral cortex into the deep subcortical masses of the brain. This is the neostriatum. It is composed of the putamen and the distinct C-shaped caudate nucleus curving over the top. Nested directly underneath the putamen is the paleostriatum, specifically the internal and external segments of the globus pallidus. As seen in this cross-section, these structures are separated from the thalamus by the internal capsule. This proximity allows for the creation of high-speed relay networks that process motor data in milliseconds. These physical clusters of gray matter serve as the hardware nodes for two opposing logic circuits, the direct and indirect pathways. The direct pathway functions as the system's go signal. It facilitates desired voluntary movements through a process of double inhibition. The sequence begins when the cerebral cortex releases excitatory glutamate to stimulate the neurons in the striatum. Once activated, the striatum releases inhibitory GABA into the globus pallidus internus, or GPI. This creates disinhibition. Because the GPI normally holds the thalamus in a state of tonic inhibition, inhibiting the GPI itself effectively cuts the brake. The freed thalamus then fires its excitatory glutamate signal back up to the motor cortex, allowing the intended action to proceed. By successfully turning off the internal braking system, the direct pathway ensures the brain can execute voluntary movement without interference. Simultaneously, the indirect pathway acts as the stop signal. Its role is to suppress the competing motor plans that the direct pathway ignores. In this circuit, the excited striatum releases GABA to inhibit the globus pallidus externus, or GPE. This triggers a second disinhibition event. The GPE normally suppresses the subthalamic nucleus. By inhibiting the GPE, the subthalamic nucleus is allowed to activate. The active subthalamic nucleus then sends an excitatory glutamate signal directly into the globus pallidus internus. This extra excitation causes the GPI to clamp down even harder on the thalamus, reinforcing the brake and preventing any signals from reaching the cortex. The indirect pathway thus creates a physiological barrier that locks out unwanted movements, ensuring the primary motor plan remains undisturbed. The balance between these two pathways is regulated by the substantia nigra pars compacta, which releases dopamine into the striatum as the system's master gain switch. Dopamine binds to D1 receptors to excite the direct pathway, while binding to D2 receptors to inhibit the indirect pathway. This dual action promotes movement by simultaneously pushing the go signal and easing the stop signal. These circuits are functionally segregated. The putamen circuit, seen here, serves as the subconscious loop for executing learned habits and motor patterns without active thought. In contrast, the caudate circuit processes conscious motor planning. It scales the intensity of our actions based on goals and environmental feedback. Together, these specific neurochemical switches and anatomical loops allow the brain to transition conscious, cognitive effort into automatic, subconscious reflex. The clinical importance of this delicate balance becomes clear when we observe what happens when the dopamine switch fails. Parkinson's disease is the anatomical degeneration of the dopaminergic neurons in the substantia nigra. Without dopamine, the direct pathway is understimulated, and the indirect pathway becomes overactive. This shifts the system into a state of severe, unopposed thalamic suppression. The thalamus is essentially locked in the off position. This physiological shutdown produces the clinical symptoms of the disease: lead pipe rigidity, an inability to initiate movement known as akinesia, and a general slowness called bradykinesia. Parkinsonian rigidity is a state of hyperinhibition. The motor gate is jammed shut, even though the muscles themselves remain fully capable of movement. A different failure in this same accessory system produces the opposite result: violent, uncontrollable movement. This is hemibolismus. It results from a discrete vascular lesion in the subthalamic nucleus. Under normal conditions, the subthalamic nucleus provides the excitatory drive that allows the GPI to brake the thalamus. Without the STN, the GPI loses its drive and the thalamic brake fails. The thalamus fires without restriction, causing flailing, coriform movements on the opposite side of the body. Huntington's disease reflects a similar loss of the inhibitory brake, though it is caused by the degeneration of GABAergic neurons within the striatum itself. The basal ganglia manage the final selection of movement. By maintaining a delicate tension between excitation and inhibition, they ensure our motor output is deliberate and contextually appropriate.